Way back in 2004, 2005, we had found a hypervirulent strain of Staph aureus in the hospital. So we wrote a grant to send it away for Sanger sequencing. A year later, and about 70,000 pounds later, we received our sequence. Fast forward 10 years, I can do the same work in less than 24 hours for less than 500 pounds. We really think that in the future, sequencing can go all the way and solve some of the challenges that we have.
I can't help but wonder how many new explanations we're going to have for some of these complex diseases that we don't understand right now.
Alzheimer's disease affects millions of people worldwide, over 5 million people in the US alone. We need to be able to have the power to say that a specific variant is associated with disease. The dream would be to identify a mutation, to be able to suggest a drug or design a drug that targets that mutation, and then to be able to slow or stop disease.
The disease that I work mainly on is a disease called ankylosing spondylitis, and we have only just, through sequencing, in the last couple of years worked out how it operates. I think we're going to see that occur across most common immune-mediated diseases over the next 5 to 10 years, and that will be incredibly exciting because you'll then be able to work out what was the environmental agent that triggered the disease, and then that raises the possibility that you might actually be able to prevent the diseases altogether.
Here at Johns Hopkins University, we're trying to develop new tools, new methods that use the latest sequencing technologies to explore human disease. I'd like to take a single cell. I'd like to just be able to sequence all of the DNA from that cell, all of the RNA from that cell, and all of the protein from that cell, and characterize that exact cell completely, but do that on millions of cells from a given sample to really understand everything that's happening. Just do that just for like per person.
6 years ago, I set up a genomic facility in the Gambia because we had none. I would like to see sequencing decentralized. If we have more sequencing available, especially in our region, in low or middle income countries, not just in Africa, it will save lives.
I got some grant to train people in Southeast Asia and also train people in UK to do fungal genome sequencing. If we can train medical students, for example, they can learn from the beginning to the end. And then they can train other people also. This is what I hope in the future.
I remember at school, I was really grateful to have the opportunity to use the MinION. So many people my age don't get that opportunity, and I think at GCSE level, if it was introduced, it would inspire more people to get involved with science. It definitely inspired me to go into medicine.
Rare diseases is a big challenge. We would like to finally get the diagnosis in 100% of cases. But we need informatics, we need genetics, we need biochemistry, biological and medical doctor, and all have to work together. I think that this way we'll get fantastic results.
My hope is that in the very near future we can really sequence every patient, any species, microbes from your gut or from the environment, and democratize sequencing for everyone and for any application.
These sorts of ideas sound like science fiction, but they're not. They really are possible. Predominantly, it's about building the healthcare systems around genomic medical services. So all of our healthcare apparatus and treatments are actually focused on people with established disease. Genomics is going to flip that on its head, so we're going to get to the situation where it is actually possible to be preemptive and focus on early disease or even before people develop disease. And wouldn't it be an exciting outcome for us all?
I think what we've achieved so far, we need to just keep going, and it will make a huge change. The future is not that far, and the accessibility of sequencing will be fantastic.
Sequencing can help us solve some of the biggest challenges that mankind are facing now, whether it's climate change, it's biodiversity, antimicrobial resistance, or new emerging pathogens. Sequencing will be involved in solving all of these problems.